All right, so now we're going to look at the exogenous antigen process. So this is where the antigen is actually presented and created outside of the cell rather than inside, right? And so if we remember from before, exogenous is referring to which major histocompatibility complex. So this is referring to involving the MHC class 2, right? And so what were the MHC class 2? This is referring to all of the helper T cells, right? So all of the antigen presenting cells that are present, right? And the specific cell type that comes in and binds to this cell is the helper T cells, right? So all the APCs, whether it's the dendritic cells, the macrophages, the B cells, all express this MHC2 complex, and they come in here, right, and bind to this helper T cell, right? So if we show the actual process, so if I draw a cell here, okay, so I have my cell here, right, and so we said the antigen comes from the outside, right? So you have this huge antigen coming in into the cell. So endocytosis takes place where the actual antigen that we have here that we've created comes into the cell, right? So this comes into the cell now, and it enters this little vesicle called the phagosome, right? So the phagosome comes in. You have all these digestive enzymes inside the phagosome that begin to break down this antigen, right? So you have this phagosome now, and you have these little antigen particles that are present waiting for binding. On the other hand, on the inside of the cell here, we're looking specifically again at any kind of dendritic cell, macrophage, or B cell, right? You have this region here called the ER, endoplasmic reticulum. So the rough ER specifically is responsible for the generation of protein. The MHC is a protein. And so now this ER is generating these little protein molecules here, these little receptors, right, in order to bind to these specific antigens, okay? So specifically, which one is this? We're talking about the MHC2, right? Specifically in order to bind to this helper T cells. So now these vesicles lyse, right? And now, excuse me, not lyse. They fuse together. They fuse together now with the ER, right? And with this ER combined, you have this new structure. You have the ER combining with the vesicles here, where all of the antigens now are binding, right? So all the antigens now, specifically here, let's just do one for the sake of show, bind to this receptor, this MHC2 receptor, right? And then finally, after this binding takes place, you have the process of exocytosis that's taking place now, where now the remaining membrane comes in and binds with this receptor here, right? And now you have this MHC receptor, so MHC2, displaying its antigen for further processing by the helper T cells, right? And so again, there's a little different, right? So all the antigens that are in order for the display comes from the outside. That's why it's called exogenous antigen process, which involves the MHC2 complex of the cell types that are specifically the antigen presenting cells, which bind specifically to the helper T cells.